This is the citric acid cycle. Glucose is broken down into pyruvate molecules, which are then sent to the citric acid cycle. The citric acid cycle takes place in the mitochondria matrix. There are 8 steps to the citric acid cycle, or otherwise known as the Krebs cycle. The first step is the acetic acid subunit of acetyl-CoA is combined with oxaloacetate to form a molecule of citrate. The acetyl coenzyme A acts only as a transporter of acetic acid from one enzyme to another. After step 1, the coenzyme is released by hydrolysis so that it may combine with another acetic acid molecule to begin the Krebs cycle again. The citric acid cycle molecule in step 2 undergoes isomerization. The hydroxyl group and an hydrogen molecule are removed from the citrate structure in the form of water. The 2 carbons form a double bond until the water molecule is added back. Only now the hydroxyl group and hydrogen molecule are reversed with respect to the original structure of the citric molecule. Thus, isotrate is formed. In this step, isocitric molecule is oxidized by an NAD molecule. The NAD molecule is reduced by the hydrogen atom in the hydroxyl group. The NAD binds with a hydrogen atom and carries off the other hydrogen atom, leaving a carbonyl group. This structure is very unstable, so a molecule of CO2 is released, creating alpha-ketoglutarate. In this step, coenzyme A returns to oxidize the alpha-ketoglutarate molecule. A molecule of NAD is reduced again to form NADH and leaves with another hydrogen. This instability causes a carbonyl group to be released as carbon dioxide, and a ester bond is formed in its place between the former alpha-ketoglutarate and coenzyme A to create a molecule of succinyl coenzyme A complex. Step 5. A water molecule sheds its hydrogen atoms to coenzyme A. Then a free-floating phosphate group displaces coenzyme A and forms a bond with the succinyl complex. The phosphate group is then transferred to a molecule of GDP to produce an energy molecule of GTP. It leaves behind a molecule of succinate. In this step, succinate is oxidized by a molecule of FAD. The FAD removes 2 hydrogen atoms from the succinate and forces a double bond to form between the 2 carbon atoms, thus creating fumarate. An enzyme adds water to the fumarate molecule to form malate. The malate is created by adding 1 hydrogen atom to a carbon atom, and then adding a hydroxyl group to a carbon next to a terminal carbonyl group. In this final step, the maltate molecule is oxidized by an NAD molecule. The carbon that carried the hydroxyl group is now converted into a carbonyl group. The end product is oxaloacetate, which then can be combined with acetyl coenzyme A and begin the Krebs cycle all over again. In summary, 3 major events occur during the Krebs cycle. 1 GDP is produced, which eventually donates a phosphate group to ADP to form ATP. 3 molecules of NAD are reduced, and 1 molecule of FAD is reduced. Although 1 molecule of GTP leads to the production of 1 ATP, the production of the reduced NAD and FAD are far more significant in the cell's energy generating process. This is because NADH and FADH2 donate their electrons to an electron transport system that generates large amounts of energy by forming many molecules of ATP. And this is the citric acid cycle.